Welcome everybody in the room and on the webinar to this month's Knauss Brownback seminar series. We have two great speakers today, so I hope you stay tuned for the entire hour. We'll be taking questions in the room and via the chat box on the webinar, so start thinking about some questions. Emily Parker is our first speaker today, working in the Office of Habitat Conservation for NMFS as a policy analyst, and she comes to us as a Ph.D. candidate in environmental engineering from UC Irvine. She earned her BS in environmental science from UCLA, and not only does she attend school in California, but she was also born and raised there. So she brings a unique perspective with her PhD work, and we're excited to hear her talk, Stormwater, Biofilters, and Kangaroos, Investigating Green Infrastructure Down Under. But first, a fun fact about Emily. I think I can safely say that Emily is the only Knauss Fellow this year who is an accordionist. So she played the accordion for 10 years, and I'm sure she'd be willing to showcase her skills. So without further ado, Emily.
Okay, thank you, Amy, for that introduction, and thank you, everyone, for being here. So I will launch right into this. So as a graduate student, I was lucky enough to be a part of an NSF-funded PIRE project, which is Partnerships for International Research and Education. So this particular PIRE was a collaboration between Southern California universities and universities in Melbourne, Australia. And as well as universities, we collaborated with other stakeholders. Since we were focusing on water, we collaborated with local water districts, NGOs, government agencies, and businesses and other engineers. And the reason we brought these two regions together, Southern California and Southeast Australia, the Melbourne region, because both of these regions have significant experience with drought. And so what we're trying to do is take what Australia learned from their drought experiences and bring it to Southern California. So one of the technologies we were interested in was green infrastructure. So what is green infrastructure? It is not this. So these are two pictures from Southern California. You see there on the left, pretty yucky looking flood control channel. And so that, what green infrastructure, sorry, this is not green infrastructure. So what you're seeing here is an example of how engineers are good at putting in pavement and impervious surface and moving stormwater as quickly as possible to prevent the risk of flooding, which is important. But what that also leads to is very uninhabitable looking spaces, not habitat friendly. And also, we are very efficiently conveying that polluted runoff directly to our coastal oceans, to our local streets. We're bringing this runoff to our streams. That means not only are we adding pollution, we're also losing out on an important freshwater resource that California could use. So on the left, I have an example of what I just showed. That is all these impervious surfaces not allowing for groundwater infiltration, not allowing for green space. And then on the right, we have our example of green infrastructure. So these are pervious surfaces that are allowing more green space in the city, but they're also allowing us to slow down the way that runoff is moving, giving it time to percolate, or giving us opportunities to capture that runoff and reuse it for other purposes. So green infrastructure can be a variety of things. Here we have a green roof, we have some pervious pavement, we have some bioswales, and all these different examples of green infrastructure also provide human and ecosystem co-benefits. And so I'm not going to talk through all these, just want to show you that they help us with pollutant removal, they help us provide habitat for pollinators, can help with flood risk. I'm going to be focusing on one specific co-benefit, and that is pollutant removal. And I'm going to be talking about it in the context of biofilters, one specific type of green infrastructure. So this is a general schematic of what a biofilter might look like. your stormwater runoff comes into the top of the inflow, enters the ponding zone before it percolates by gravity through the filter media. And then on the left, we have a traditional outflow at the bottom. On the right, the outflow is elevated to allow for a submerged zone. These are just two different biofilter designs. And so biofilters can do a lot of things. They help us with removing nutrients, heavy metals through various processes, removal of contaminants, pathogens, suspended solids. Biofilters are pretty cool. So I'm going to be talking specifically about fecal indicator bacteria removal. So quickly, just to tell you what fecal indicator bacteria are. They are non-pathogenic bacteria, high concentrations in feces, and they're used as proxies for pathogens. Enterococcus is used for marine waters, and then E. coli in fresh waters. So fecal indicator bacteria are not perfect proxies for pathogen inputs because they may come from non-human sources or they regrow in the environment, but they are still incredibly important from a regulatory framework, and that's why I've focused on FIB. And as you can see, FIB concentrations in stormwater and in dry weather runoff are 1 to 2 orders of magnitude higher than the water quality criteria. So if we exceed this criteria, that's when beaches are closed to swimming, and runoff is one important source of those FIB. Important from a standpoint of illness, recreation, and cleanup, FIB are also expensive, so that's why I'm talking about them. All right, so biofilter, we've got the bio component. That might be the plants, the animals, bacteria that are living within the biofilter. They all contribute to those pollutant removal processes. Then we've also got the filter portion, which is referring to the filter media, typically a sand or loamy sand compost combination. So let's just start by talking about the filter. As environmental engineers, we want to apply the simplest theory we can to try and predict how that filter might apply to FIB. So today I'm talking about clean bed filtration theory. Very simply, this is that the sand grains are acting as collectors, and the FIB are going to attach to those collectors as the stormwater runoff is moving vertically through. But this is a theory, so it has a lot of assumptions associated with it. For example, it assumes that the biofilter is always completely saturated with runoff, and that it is constantly flowing, which of course is not true because it's not constantly raining, so there's not constant rainwater moving through that system. It assumes that the collectors are spherical, and of course sand grains are not perfectly spherical. It also assumes that all the collectors are clean, that is, there's no previously deposited particles attached to them. So as you can imagine in biofilters are not clean because we're putting polluted runoff through them. So the question here is this theory at all useful for predicting what happens in green infrastructure? And if it is useful, or if it's not useful, to what extent do other factors explain what that theory cannot? So then we get to talking about the biology of the system, the hydrology, the ecology. How helpful is that? So I'm going to outline the experiments we did here. First, I'm going to talk about experiments that we conducted, and then a second set of experiments previously published that we also pulled into our analysis. So these are our experimental biofilters. So they're constructed in big PVC pipe columns. You can see in the schematic here we have a layer of fine sand, coarse sand, and then a drainage layer of gravel at the bottom. So that's the filter media, the fine sand. We had 3 biofilters that were planted with Carex suppressa, which is a very common biofilter plant in Australia. 3 of them were unplanted, so we had 6 total. And we also had an overflow drain at the top to allow us to keep a constant level of water in the ponding zone. And so this picture is from when we did these experiments in 2015, and we allowed the columns to grow for about 9 months. And so I just wanted to show that the plants were happy and they did grow a lot within that year. So, and the way these experiments were conducted, the first thing we wanted to do was just know how long it takes water to move through the column. How long is water spending in there? And so to do that, we conducted pulse experiments with simply salt water, so we could measure salt water going in, salt water coming out. We added a pulse of salt water to the top of the column, and then measured it coming out of the outflow, like in the middle picture there. We wanted to conduct these as saturated, constant flow experiments to align with the underlying assumptions of that theory that I mentioned. So we set up a system, you can see on the right picture, to allow for constant water flow to all the columns. And just to give you an idea of what these results look like, these are, we call them residence time distributions, really just telling you how long water is spending in the column. So each of these plots corresponds to one of the biofilter columns. The x-axis is just time, residence time, and then the y-axis is the distribution. So for example, in column A, you'll see a peak around 30 or 40 minutes. Just means that most of the water is spending 30 or 40 minutes in the column. So just allowing us to get that basic characterization of what the water is doing. Once we have that, we want to know what the FIB are doing. So this is where it gets fun and exciting, and we start putting sewage into the columns instead. So you'll notice that we're wearing face masks when we're doing the sampling here. So same thing, we're adding a pulse of sewage to the top of the column, and then measuring, collecting what comes out at the bottom of the column, and then quantifying how much bacteria are in those, so we can see how much bacteria the biofilter was able to remove. And then the other experiments I mentioned, these were published, but they were conducted in a pretty similar manner, and we just used those to increase our sample size, but also to expand our framework, because this set of experiments had multiple plant types. So we were, instead of just looking at the one plant type, we were able to look at multiple different types of plants, see if that had an effect. And these were not conducted as fully saturated experiments. They were conducted more realistic of what you would see with rainfall and then a dry period and then rainfall. So these are a little more realistic, but less aligned to the theory. So it allows us to compare. So those are the main differences. 196 of these were included. All right, so again, the question was, is that theory useful at all? And how can we use other factors to explain what that theory might not be capturing? So the first thing we do, let's compare the FIB removal rates that we observed in our experiments, and then compare them to what the theory predicts. So in this plot, your x-axis is what the observed FIB removal, y-axis is what the theory would predict, and then the different colored markers, green and black are the experiments we did, all the red markers are from that separate set of previously published experiments. So you can see for the set of red markers, not great agreement, but then when we look at our own experiments, we do see better agreement. And one explanation for this is that our experiments were conducted under conditions that more closely align to the theory. So now what if we take into account some other factors? We want to know, did the type of plant affect it? The fact that the biofilter was built with an elevated outlet and had a submerged zone, did that matter? Did it matter if it was saturated or not? So this is where we pulled in multiple linear regression. So here we have the same axes, the observed values on the x-axis, and then the y-axis is our multiple linear regression model. Here we just included the additional previously published experiments. So we see much better agreement with the red markers, not great with the green and black. And the additional factors we pulled in were the age of the biofilters. So the biofilters mature, their performance changes. Antecedent dry period, how many days was that biofilter dry before it received some stormwater? Design of the biofilter, if it had a submerged zone or not, and then the type of plant. And we do this one more time, but here we're going to combine all the experiments. So here we're, again, same factors, but with all the results. And so we're seeing better agreement between what is observed and that is what is predicted by the theory in conjunction, taking into account those other factors. So is clean bed filtration theory useful? It explains about 31% of the variance. It is useful when we consider it with these other factors. So antecedent dry period, again, how many days was the biofilter dry before receiving stormwater, counted for about 14%. Study effects, so the difference between our experiments and the other ones, was 8%. The age of the biofilter explained about 7%. And then presence or absence of shrubs, specifically 6%. So the other plant types didn't come up as important, only shrubs. So individually, none of these factors tell us much, but collectively, we start to be able to explain what's going on in this biofilter. And so this is a really unique way to take engineering design theory, this clean bed filtration theory, and then combine it with the other piece, the biology and the ecology, to try and actually predict how these biofilters perform. And I won't talk through all of this, just wanted to point out that when we start having these frameworks where we're able to predict what happens, then we can start looking at the variables like infiltration rate here on the x-axis, FIB removal is on the y-axis, and we have more removal the lower to the bottom you are. And each of these colored curves represents a different biofilter system, either in California or in Australia. And we're able to see where is the optimal, for example, infiltration rate. And we can, as an engineer, we want to be able to design for the optimum, and we can see what might happen if we started at a higher infiltration rate, and then the biofilter started clogging and moved toward a lower infiltration rate. How is that going to affect the performance? So if you enjoyed that, you can go read this paper that was just published. I briefly touched on some of what was in here, but there's a lot more to read and enjoy if you're interested. And now I'm going to switch gears a little bit and just talk about working in Australia, show you some pictures, just have a little more fun. So one thing that's very clear about being in Melbourne is that there is a different mindset about water. So new developments all have rain tanks attached to their houses. And when you walk through neighborhoods, you see people put signs in their front yards saying, I use water from a rain tank. People paint their rain tanks and personalize them and take a lot of joy in their rain tanks, which is fun. We see signs that toilets are flushed with sustainable rainwater. Every single toilet I saw in Australia was a dual flush toilet, had 2 different buttons. I did not see anywhere in Australia a normal toilet like you would here. And the signage related to these systems is pretty evident, so you'll see a lot of efforts to educate and get people excited about these types of systems. So building rain gardens, biofilters, any type of green infrastructure is sort of becoming a built-in way of life in Melbourne, I would say. Then this is just fun stuff. Australia is really pretty. This is the Great Ocean Road. So just really pretty scenery that I got to see on the 2 days when I wasn't in the lab there. Other cultural experiences. Anyone here been Vegemite? Yeah? So if you're having your first Vegemite experience, don't do what a lot of us did and spread it on your toast like peanut butter. It is not like peanut butter. In this case, you want to, I would say a lot of butter, and then the thinnest possible layer of Vegemite. So thin you can't even see it. And only then is it tasty. What about Tim Tams? Anyone had Tim Tams? Yeah, so those are I'd say way better than Vegemite. Find some Tim Tams, those are fantastic. And then, of course, the local wildlife. So koalas are cute and fun. Kangaroos are fun to pet and feed. come hop around. We all thought this was great until we went into the grocery store the next day. But you know what? It's actually delicious. So I'm going to stop there. Say thank you to all my co-authors, everyone involved in the PIRE project, everyone I've worked with. Thank you for listening, and I will take any questions now.
We have one question from the chat.
Okay.
From Christopher. Based on the green infrastructure diagram, it looked like you get an increase in groundwater export into coastal waters. Does this pose any problem?
Let me go back to the slide. Really just saying that we're getting more groundwater percolating downward into our groundwater storage. Not saying that it's actually specifically going anywhere. And in some cases, it's hard to implement green infrastructure because the soil is not suitable for recharge, or the water table is already too high, you can't recharge there. So that's all, but all we're really trying to show here is that green infrastructure allows for increased recharge if you're in a region that it works. So,
or do you know, are these like in place in California today?
Yes. So the question is whether that, where is the runoff going next? Yes. So are we, in some cases, it's you can capture it and reuse it for something else. Like say you want to use it for irrigation or toilet flushing. The policy framework and regulations have to be there to allow that to happen, and strict standards about how clean the water is. Or that water might go back into the storm sewer system and be discharged back to the ocean.
Do these biofilter systems usually be placed in cities? Are they on buffer zones between rivers, or are they kind of just scattered throughout the concrete cities where they usually?
So they can be very decentralized. So if you'll walk, like if you're walking downtown by the National Mall right in front of the EPA building, they have several of them built right into the sidewalk there in front of the EPA building. So a lot of times they would be along the street where you would see in the gutter stormwater going into a storm drain. Instead, you'll see that stormwater being funneled from the gutter into one of these systems. So since they're vertical systems, they can have a pretty small aerial footprint. So they can be scattered about pretty much everywhere. But there are also larger scale systems that, for example, collect all the runoff from a section of highway, and they're much larger along the side of the highway. But they can really be all over. That's probably a much better use of clean bed filtration theory, because you're talking about spherical collectors. Yeah, yeah.
Have you read about any nature flows?
No. I like it. Yeah. So the question was about biofilter maintenance. Maintenance is a big deal, and it's still one of the big unknowns, is that as green infrastructure has become more popular, as you know, now some of these systems have been around for 10 or 15 years, and the question is, are they still performing as they were when they were installed? What kind of maintenance do they need? So it's still kind of an up in the air thing, and you know, at some point, do you have to completely reinstall the system? The filter is just too clogged. But it seems so far as if you can kind of do a scrape of the top layer, maybe the top couple centimeters every year, every couple of years, and that gets rid of a lot of the particles that are doing that clogging, and then maybe come back and adding more, doing that scrape. So the question was about biofilter depth, and yeah, our experiments are pretty typical of what you would see. Around a meter depth is usual. So the question is, what's the benefit of having a submerged zone, like you see on the right here? And that is to create an area that is saturated anoxic to promote denitrification. Thanks, everyone.